You know, there's a whole invisible world living on every single surface you touch. Your keyboard, your doorknob, everything. But how do you actually see something that's microscopic? Well, today we're going to dive into the clever scientific techniques that make this hidden world totally visible. Just think about the phone you're probably holding in your hand right now. It is home to a bustling little ecosystem of microscopic life. I mean, some studies show it can have 10 times more bacteria than a public toilet seat. Seriously, but what exactly is living on that screen? We're about to walk through the scientific process for finding out, step by step.
All right, first things first. Before we can even think about finding these microbes, we need to understand a little bit about what they are. So let's go ahead and meet our tiny subjects.
So, the whole story starts with this huge group of organisms called prokaryotes. The key difference between them and, say, us is actually pretty simple. Their cells don't have a nucleus. All their genetic info just kind of floats around inside. You can think of it as a more ancient, streamlined design for life. And the most famous prokaryotes of all? You guessed it, bacteria. These single-celled powerhouses are literally everywhere, in the soil, on your skin, and yep, all over your phone. Now, we often think of them as germs, but most are totally harmless, and a lot of them are even helpful. The bottom line is, they're just an unavoidable part of our world. Okay, so we know what we're looking for. The challenge now is getting these invisible little microbes to actually show themselves. And to do that, we have to create the perfect environment for them to grow, until there are millions of them.
So here's the puzzle, right? How do you take a single invisible bacterium and make it visible to the naked eye? Well, the secret is to give it a 5-star hotel with an all-you-can-eat buffet and just encourage it to multiply into a massive, visible colony.
To do this, scientists use something called a culture medium. This is that all-you-can-eat buffet for bacteria. It's a special substance, usually a gel, that's just packed with all the food and nutrients a microbe could possibly want. It lets them multiply over and over until their tiny community becomes a whole visible city. Now, you've got a couple of main options for this microbial buffet. You might have even seen simple gelatin-based ones in a science class, but in professional labs, the gold standard is an agar plate. It provides this really firm, nutrient-rich surface that's just perfect for bacterial growth.
Okay, so we've got the food source, but there's one more absolutely crucial element for success, and it's this exact number, 37 degrees Celsius. So why? Why is this specific temperature the magic number? It's because 37 degrees Celsius, that's 98.6 Fahrenheit, is our own body temperature. The bacteria we collect from the surfaces we touch every day are already adapted to thrive in our warmth. So by putting them in an incubator at this temperature, we're basically giving them the perfect tropical vacation, and they respond by multiplying like crazy. Okay, so we've got our theory, our agar plate, and the right temperature. It's time for the most important part, actually collecting the sample. In science, careful technique is everything, so let's break down the 5 key steps to get it right. First, sterility is absolutely key, so always use gloves and a fresh, sterile swab. Second, rub that swab firmly on the surface in a zigzag motion to make sure you get a good sample. Third, and this is important, immediately transfer that sample to the agar plate. You don't want anything from the air getting on there. Fourth, label your plate with everything. Unlabeled data is useless. And finally, a really clever but critical step. Place the plate upside down in the incubator. This stops any condensation from dripping down and messing up your experiment.
Look, the whole reason we go through this careful process is to be sure of one thing, that whatever grows on your plate is from the surface you tested and not from your hands or the air. It's all about getting clean, reliable data.
And now for the really exciting part. After a couple of days in that nice, warm incubator, you open the door, and suddenly the invisible has become visible. But what exactly are we looking at? Let's decode these results.
Every single dot you see on that plate is a bacterial colony. And that's not just one bacterium. Oh no, it's a massive city of millions, sometimes even billions of cells. And they all grew from just 1 or 2 original bacteria that landed on that spot from your swab.
For a microbiologist, each one of these colonies is a clue. They'll start by describing its physical traits. What's its shape and color? Is the texture shiny or is it matte? Is it flat or is it raised up? How big is it? These simple observations are the very first step toward figuring out what specific type of bacteria you're looking at.
So, let's put this into practice. Imagine you swab your keyboard, and a lab analysis identifies one of the colonies as Escherichia coli, which you probably know as E. coli. What does finding that specific bacterium on your keyboard actually mean? Well, since E. coli is primarily found in the gut, discovering it on your keyboard is a pretty clear sign of contamination. It almost always points back to poor hygiene, specifically not washing your hands properly. It's a perfect, if kind of gross, example of how this invisible world directly reflects our daily habits.
So what's the big takeaway from all this? This whole process, from the first swab to seeing that final colony, is way more than just a neat science experiment. It really highlights the importance of careful work and, just as important, relying on good information.
Whenever you're dealing with any kind of biological materials, even microscopic ones, being accurate and responsible is just non-negotiable. Careful handling, precise labeling, sharp observation, that's what makes scientific results trustworthy in the first place.
And you know, being a smart consumer of science is just as important as doing good science. When you see some headline about bacteria on your phone or a new superbug, make sure you're getting your info from reliable sources. Organizations like the CDC and the WHO, or peer-reviewed studies. That's where you'll find the facts, not the fear.
So now you know the fundamental principles for making the invisible world visible, from collecting a sample, to giving it a home, to reading the results. You've basically got the map to a hidden world that's all around you, all the time. The only question left is, now that you know how to look, what will you discover?